Okay, so welcome to this fourth presentation, first series of meetings where we've been listening to the grantees. It's been a good sequence of 3 meetings, and this is the fourth. So we're looking forward to the presentations we'll hear today. Again, it's around 15 minutes presentation, 5 minutes Q&A. Yeah, and we're looking forward to hearing from all of you. So without further ado, Ahmed, over to you. And as you present, you can keep your video open, video on. That will be good for us to see you as you speak. But go ahead, Ahmed.
Yeah, good morning, Prof. And good morning, everyone. My name is Ahmed Kehinde Usman from Ahmadu Bello University. I'm a PhD student in Applied Geophysics Unit from the Department of Physics. My title of the presentation is Application of Spectral Analysis Technique and Machine Learning in Evaluation of Geothermal Energy Resources Potential in Northeastern Nigeria. Nigeria, as a country, we are actually facing the challenges of insufficient electricity generation. And this actually has a setback on the technology and the economy of the country. A lot of researchers have worked on the geothermal energy to at least to serve as a complement for the normal hydrothermal that we have already been using, hydro power generation that we have been using. So the geothermal energy is in a short term, is just the use of heat energy within the Earth's surface. And it has been documented that the deeper we go down the surface of the Earth, the hotter the temperature becomes. So this geothermal energy, it has been reported that it is a very clean energy service because the emission of greenhouse gases they emitted, they are very small compared to other sources. And this will be a very great advantage for Nigeria as a country, very rich in oil and gases. If this energy is being developed, this will generate a very great, it will generate revenue for the country and also generate a kind of job creation for the youth and allow the youth to actually display their own talents instead of depending on other nations of the world. Spectral analysis that was actually deployed in this work is just the analysis, or let's say simply the calculation and interpretation of a spectrum of potential field data. And we use this analysis to calculate what we know as Curie point depth, the geothermal gradient, and heat flow. The Curie point depth is just the depth at which a ferromagnetic material actually loses its magnetism and changes to paramagnetic materials. And geothermal gradient is just the rate at which the temperature increases with depth. This actually shows as how heat flows from the interior to the surface, and heat flow is simply the flowing of heat from the Earth's interior to the surface. For this work, the justification is that, as I said earlier, insufficient electricity supply, this has actually driven the technology development advancement in the country, and a lot of emphasis on energy derived from crude oil, which has a lot of side effects on the environment and people within that region. So this energy, this geothermal energy is actually a very clean and sustainable energy. And this will actually allow the society to have access to, access to electricity, at least if it's not more than 20 to 15 hours, instead of like some regions having 3 to 4 hours, some less than 10 hours, and so on. So this will actually be a very good advantage to boost the nation's economy and thereby create a feeling for youth, also in terms of job creation and so on. And also in the region where I am conducting my research, there are a lot of geological evidence for the geothermal energy in the region. There is what's known as hot springs and basins within that region. And there is the tertiary continuous rocks. Also, we have a report of uranium mineralization in the area. So this work is actually very necessary to give a full analysis of the geothermal potential in the area. And the aim is actually to evaluate the geothermal energy potential within northeastern Nigeria, which can be exploited in order to serve as an alternative sources of energy using spectral analysis. And the objectives I'm working on for this work are to determine land surface temperature.
No,
and
okay.
then,
You click on the link.
which link?
No, go ahead. I think that was a mistake. Go ahead.
All right, sir. So the objectives, as I said earlier, they are to determine the cooling point depth, geothermal gradient, heat flow, and so on, and also to delineate lineaments, and finally to identify areas of higher geothermal potential. So for this particular presentation, the progress, I will be giving reports on the land surface temperature, cooling point depth, geothermal gradient, and heat flow. The lineaments and the area of the artificial intelligence will be covered in the next presentation. And this is just about the literature review concerning the area. Some of the literature review I have in my work. So some researchers have worked on this particular geothermal energy potential in the area. And I have some comments concerning their work. So these are some of the comments that I have. Now, the methodology used. The area covered the latitude of 9 degrees 30 minutes to 14 degrees when it comes to latitude, and the longitude is 9 to 14 degrees 30 minutes. And then the rocks in the area, we have the report of 3 different rocks in the area according to Jallo 2015. Also, for this presentation, I will be giving reports on the satellite imagery data in which I used Landsat 9 level 2, gotten from United States Geological Survey sites, and also the high resolution aeromagnetic data collected from Nigeria Geological Survey Agency. So for this aeromagnetic data, a total of 85 sheets were actually bought and processed. So this is a general TMI map of the area collected from the NGSA after putting into OASIS montage. So the TMI was given to me after payment. So this shows the grid for each sheet. And this is the normal, the TMI map. And this gives us the regional map of the study area. This was gotten from the main TMI, the regional and the residual. This is our residual map on which we actually worked on to generate our cooling point depth and geothermal gradient and heat flow. So the data were processed using OASIS montage software, ArcGIS, Excel, so far. And the results I have presently are all these. Now, for the LST, USGA, they gave us some formulas to work using raster calculator from ArcGIS. So I calculate TOA, which is a top of atmosphere, and BT, brightness, temperature, NDVI, PV, emissivity, and finally the LST, using all these formulas from the raster calculator in ArcGIS. And also from the literature review, the estimating cooling point dates, I need to use this formula ZB, which is our depth to centroid with magnetic source, and then the Z0 and ZT. The ZB is the cooling point depth. Sorry, the Z0 is the depth to centroid, while ZT is the depth to the magnetic anomalous body. So this Z0 and ZT were calculated from the map, and this were put into Excel to calculate our ZB. And from there, I calculated my geothermal gradient using this formula. And finally, the heat flow. This is just my my geothermal gradient of the Curie point depth. And then this K in the heat flow is given as 2.5 from literature review. Now the results for the digital elevation model of the study area, which is just showing like the general elevation of the area, and we can see that the region in general is actually like a very high region. You know, the northeastern Nigeria, like a very the desert. So we can see like 52 to 1752. And you can see this map showing the vegetation in the area. You can see they're very low because, you know, the area they are more like desert area. Then this map of the band 4, because from the NASAT 9, we have different bands. So we use band 4 and 5 for our NDVA, and also band 10 to calculate from the top of atmosphere and so on. So this is the map showing the mosaic map for all the band 4 for the region, because, you know, the region is a very, very large region. So we have different data sets. So then this is the map for band 10 and the total top of atmosphere of the area. So as it can be seen, you will see this where it's actually the values range from 0.2 to 25. And at this, and can be seen, we can see a very high region top of atmosphere around this north east, and then within the study area. Also for the brightness temperature, you can see the map and then the NDVI. So this is like normalized digital vegetation intensity of the area. So as it can be shown, you will see that for the high NDVI, it's actually recorded around this area in the northwest of the region, and also the PV, proportion of vegetation of map of the study area, and the emissivity map of the study area also. But the one that is actually working on is the LST, the land surface temperature map of the study area. From this, you can see the range from -129 and to 24. So the ILST recorded around this area here and this within the study area also. Also, this is the map of the spectrum for the for sheet 1, because I divided my own sheet. I used sheet 1 to sheet 80, so to 85 for sheet 1, and this is for sheet 2, 3, 4, and so on. Also, this is the one we use to calculate our Z to top and Z to centroid, as it can be seen. You will see this, the ZT, and this is the Z0, this for sheet 1, for sheet 2, sheet 3, 4, and so on. So I try to quote it because of the presentation. And this was actually used to calculate our Curie point depth. And after that, we exploit this, we install our software to have the control map of the Curie point depth for the study area. So this shows the Curie point depth in kilometer, and the values ranges from 10 to 52. So, and you can see this is very, actually very deep. It's very deep, because 10 kilometers is not a small distance. So we can actually record we need area that are very shallow area, as it can be seen here, and this particular region here and here. So also these are also not that very high, this green showing these green regions. And also for the geothermal gradient, this, you know, we say the what temperature divided by per depth. So we can see the region according to temperature. This one they actually need about 30. If you have 30 degrees Celsius per kilometer, yeah, it's a very good area for geothermal energy. So as can be seen from our 30 here, so we can see the region around here as this actually refilled. So we can see the very high region around this area, which is actually corresponding to the Curie point depth, very low Curie point depth here. So this area showing high geothermal gradients, and this area, and within this area too. Also for our heat flow according to researchers, they prefer for the dashboard region like desert area, they prefer like 100 and above heat flow. Though, so some research that actually depicted that less than 100 can also be considered. So if you look at our map A, the region for 100 heat flow, you can see from this 103, 97, and so on. So if you look at this area, around this southern region, southeast, you can see this region corresponding to all this, and also this region for high heat flow. Now, these are the results for the Z0.
Ahmed, I just ask you to maybe summarize.
Summarize. Yes, I'm even going to the conclusion now. Yes, sir. So this is just the table showing all the values, and then the results, just as I said earlier, the LST, you could see area of high temperature and low temperature, and the areas of low Curie point depth, that's where we are actually considering, and we need to correspond with area of high geothermal gradient and high heat flow. So the result, we feel possible geothermal potential, just the present result we have from ground and higher from ground, and the regions of good and high potentials were identified around the southern, eastern, and central parts of the study area, which serve as indications of geothermal reservoirs. So these are some of the references. So the next the presentations, so we are actually I'm going to give the full details of what we have, what will be happening in the study area. Thanks for listening.
Yeah, thank you very much for your presentation. I welcome questions. Samson, maybe you can just unmute and ask.
Yeah, thank you, Ahmed, for the very good presentation.
Thank you.
The visualizations are very, very good. I just wanted to understand what do they mean now that you have the visualizations that you were able to generate from the analysis you did using the spectral data. What kind of insights do they provide with respect to the energy sector? I'm just trying to look on the applied side of things. Yeah.
Yeah, the application actually, this geothermal energy, the equipment actually is similar to oil and gas. In essence, it's similar to oil and gas equipment. So, and as we know, there are some basins already in Nigeria that have been abandoned, and there are some that have still been discovered. So if Nigeria can actually work on this, so it will not even take much, like maybe much resources for it to develop and exploit this. So all these results, like if you look at this now, for example, this geothermal gradient and heat flow, we will need consequent researchers to actually work on this area of high potentials. So they need to actually exploit the area very well to actually locate and know the areas that can be actually work on, and give government and maybe recommend to the government to exploit. So
yeah, thank you very much. That's really very, very good, because it will save costs, because now you know the exact locations.
Yes.
Yeah, thank you very much.
You're welcome. Thank you.
Okay, I see Shegun wants to ask a question, but I had seen Samson. Samson, did you want to ask a question?
Yeah, it has been covered by what has been asked, so I'm okay. Thank you.
All right, Shegun, go ahead, unmute.
Hello, I can't hear you.
Can you hear me?
Yes, I can hear you now.
Okay, so I thank you for a good presentation. My question is, I'm just wondering, how can we apply machine learning, to be able to take, for example, if we don't have access to this data? I believe you use satellite data, which might be access to it might probably not be impossible at times. How can machine learning, help in trying to, make prediction in identifying some of these areas based on probably your result or maybe analysis you have already on ground? That's just my own concern.
So thank you, Mr. Segment, for your question. I'm really very grateful. Yeah, that question is actually going to be covered in my final presentation, because I'm proposing actually to make research of maybe probably these high regions. That's what I'm actually the kind of machine learning I'm trying to work on, shows that the consequent researchers can actually develop on. When they can pick like these areas that are the potential areas, then if I develop a particular, maybe like an equation, do you get it now? They can now maybe develop on that to see, and they will be able to use that to actually depict maybe if there are areas also within that region. If you are now working on this, there are still areas that of high potentials like these areas, but not as this. So if I have an equation, the researchers can work on that to see if the equations can also be extended to that areas. So that is the plan actually, and the kind of the proposal I have to give the research on. So probably in my next presentation, then I'll be able to answer those questions very well and maybe give the details of all this.
Okay, thank you very much. I
Yeah,
look forward to following you and getting it. That's very good.
welcome. Thank you very much. Yeah.
Right. I think we'll thank the speaker. Thank you very much, Ahmed, for that insightful presentation.
Thank you, Paul.
Forward again to future presentations.
Sir.
Can appreciate you, and then call on the next speaker, Dr. Kadege, and thank you for graciously allowing Ahmed to go first.
All right, thank you, sir. I'm very grateful, doctor. All right, bye.
Okay, so the next presentation is, I guess you'll be ready to present.
Okay. I think everybody's good afternoon, everyone. My name is Kadege. Let me see here. I want to set it to presentation format. Can you see my presentation?
Yes, we can see it, though it's not in it's not full presentation mode.
Let me do here. I can see it, right?
Yes, it looks good. If you can move from one slide to the next, then we are good.
Right? You can see all this, right? Okay.
I can see it's the first slide. So all the go ahead. You have 15 minutes.
Okay. Okay. So I'm Kadege Fue from Tanzania. I work for Sokoine University of Agriculture. I'm here to present prediction of rain-fed maize yield in Tanzania using time series machine learning models. So I'll talk a little bit. We have a team that worked on this project. So I have Anna, Betha, Donat, and Dixon who are working at our lab. And they work on this most of like collecting data, cleaning it, formatting, and all those kind of stuff. So I'll talk in detail on how we like a problem statement, out of our research objectives, significance, and all those kind of important details. And then I'll go on why do we need this research? As you know, we have been struggling with hunger across Africa, and also we have a big problem with climate change, and also a lot of things are changing, especially on how the weather is regulated. So with this research, we're looking on how we can predict and see like how climate changes can be predicted and see how the changes can be tracked so that we can have appropriate mitigation strategies. So one of the methods actually we use in Tanzania is called Water Requirement Expectation Index. And this algorithm, we worked with it in the year 2013, 2014, where we were looking on qualitative method. It's a qualitative method that says how for example, especially the cereal crop, how it performed for that single season. It looks, on the very few parameters, rainfall and evapotranspiration. But this WSI can be improved actually by using machine learning, and it has been shown in some areas, especially in Asia and Brazil and America. So what we do is ML can incorporate more data compared to what we do in WSI. So the data especially for training machine learning, the good thing is available, especially from the climate store at the Copernicus EU data store, and it's also available at the National Weather Administration from the US, and also
Have we lost Dr. Fue, or is it just me?
I
think we've lost him. I thought it was just me as well.
You cannot.
Yeah, maybe you can also just limit the video if you have some challenges. We had lost you for a minute.
Cannot hear me.
Yeah, maybe just go. Yeah, we just lost you briefly. Can you hear me now?
Now you can hear me.
I can
right?
hear you. So you can also, if the bandwidth is an issue, video, you can do away with it.
Okay, I can disable. Let me share again. Oh. You can see my slides, right?
Yes.
Okay. 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 So let me continue from where I left. So prediction of field is quite important for the government, especially to mitigate crop performance and for food security. So with this project, we're looking on using geospatial analysis to look on how we can predict maize. Another issue, so what we want to do, especially on this, is to forecast rain-fed maize. And the second very specific objective is on exploring and identifying appropriate climate parameters and yield data that can be available, especially on open data stores, and also then to develop several machine learning models and see how the performance is. So one of the key questions that we had was, what are the main climate parameters contributing to prediction of maize crops yield in rainfed agriculture in Tanzania, and looking on how the conventional machine learning models that are available and how they can predict maize yield in Tanzania. And the last thing, which is the important, and how we can use time series models now to improve prediction from the conventional machine learning models. So here we're looking on, so this, why do we need this? We want to contribute for policymakers, especially on the output. We expect this can help policymakers make evidence-based policies, and also for farmers to use as the output for study. Outputs can be used as to predict yield. They are not the ones that will be using like as the output directly, but we can implement inference engine that they can have as maybe sending messages or some kind of those inference engines. And for researchers is to suggest how we have explored, and so that the researchers can take on improving using more powerful models. So for this, we had to look on some of the very methods that have been used in yield prediction, especially in Tanzania, can be in East Africa, can be in Africa. So we looked on several models, average models, naive models, regression models, and artificial neural networks. So one of the model that is being used in Tanzania is called WSI. It has strength, like I've said, but it has limitation, especially on linear issues. And also the output is only qualitative. And most of the stakeholders now, they're looking on getting quantitative information. They don't just need excellent, very good, good, or something like that. They want more information on that. Or if it's classes, then a wide range of classes. Another models that have been used popular in East Africa, South Africa, I've seen it in some of northern part of Africa and also Western Africa, is called regression models. And they've shown they're very easy to implement, and also they can predict even before, like 6 weeks before. But most of the prediction depends on how the data is. So linearity of your data actually will give them more accuracy. If the data is much more different, or the country has very different climate change, it has different climate conditions, then probably it will be difficult to use that one. The other ones that have shown very powerful performance is conventional machine learning, such as random forest, adder boosting, gradient boosting, k-neighbor regression, random forest. These have shown very good performance, mostly in Africa, and they have been explored so much. For example, especially for nonlinear data, like agricultural data, but they have not shown very good performance when you consider time series or changes as the climate, because the climate is not constant, it's changing as we go on. So they have shown weakness on that. Another which is quite important is artificial neural network and deep learning methods. They're common around the world, especially on using like images and all other kind of data. And if you are checking, like predicting yield using satellite image data, but they are very complex, and they need computing resources, that's why they are not preferable in Africa, because and also training takes so much time, and it's very expensive in training, especially if you have a gigabyte of data to train. So what we did in this research, we explored some of the database that are available around the world, and one of the data that was very important to use is called Copernicus ERA5 land reanalysis data. This was an amazing data set that is available from the European Union, and this is the data can be provided across Tanzania, because in Tanzania we have very few weather stations, so you cannot get all the data all the places. But with the ERA5, it's reanalyzed data, which provide almost in every point of Tanzania you can get an estimate of the climate change. So this is very good, especially if you want to predict for in district level yield prediction. So we look on developing this, and we developed basic machine learning models, and then we validated these machine learning models, and also we developed what we call stacking random forest regression. And then we trained the model and looked on different evaluation techniques. For example, mean absolute, you look on mean squared error, because this was quantitative research. Now we looked because this model was based on district-wise, we had to look on several data sets. We looked on how we can predict, for example, 2016 data by using data from 2005 to 2015, and also we tried to train the model to predict 2017 by using 2005 and 2016, and also 2005 to 2017 to predict for 2018 data. So one of the problems actually we found, the data set, especially for yield data, it had so much problems. So we had to call the Minister of Education, Minister of Agriculture, so that they can explain to us what could be the problem. They told us what could be the problem and why the data had so much deviation. So we had to remove some of the district that we thought probably this district had very bad data. So in Tanzania, we have almost 183 districts, but we had only to consider 94 that we thought the data were much close, or the data was somehow clean that we can use for prediction. So we had to remove so that our models can work. So with this, it was good for us to get the data. So another thing is climate store. You can get the climate store data, Copernicus climate store, where you can get the data set. Era 5 reanalysis data, and this data can start from 1950 because it has been modified a little bit to make sure like it obeys physics law. So it is somehow clean data with the experts from weather, the experts of climate. So it's a very good data set to use, and it has shown efficacy, and it has been trusted by most of the scientists, climate scientists from Europe, Asia, America, and Africa. So it's a very good data to use. So these are the weather data that we considered. So we considered almost like 10 data set. Like you can have wind component data, temperature, evaporation, runoff, total precipitation, soil temperature at level 1, 2, and 3, surface net solar radiation, evaporation for vegetation, and then volumetric soil water layer, 1, 2, 3, all the layers that are quite kind of very important for prediction. And by using this data, we had to compute yield data from the production against hectares. And from that data, we had to get all the seasons, 13 seasons. So we had to process 13 seasons. And major location where maize is planted was estimated by using normalized different vegetation index. We had a problem, like when you want to predict accurately yield, you really need to know where the farms are. You need to get coordinates of those farms. But in Tanzania, we don't have such data. So what we did, in order to avoid to use temperature or to use volumetric data from an area which is urban, we had to use normalized different vegetation index, where we had to run a code that can show us exactly where plantation occur. And we looked on most of the time where the season we plant. So we had to look on those areas and the bit or get the information from that data. And then later on get that data for each month and for each year. And by using that data for each district, it was possible to know exactly where probably their plantation or the areas that you can look on that is a good temperature, or you can estimate the temperature and rainfall, which is good for plantations. So not looking on all areas of the district. Another thing is,
Daktari, your
Yes.
time is up. Maybe you summarize.
Okay. Let me summarize quickly. So we had to look and clean the data. So look and cleaning data, make sure like the data is balanced. That's when we removed some of the district. And then we had to check on missing data. And then we had to standardize and then train the model for each year, and then find out who, by validating for each year and see how the model was performing and how it was looking. And then so we looked on each year, we had to plot the graph and see like how the data is performing. And also for 2017 prediction, and also look on 2018. We are trying to see like how the model is performing different years and see how it is performing, maybe looking how uniform it is. And then we had to plot the data, how it was, how the inference, how the training was occurring, and how the score was gotten. And also we look on fitness of the model, and then looking on how the prediction comparable to the score and fitness of the model for 2017, 2018. And so that was for machine, what we call conventional machine learning. So we had to look on time series, and this time series we look on long short-term memory. And we trained the model and compared it with LSTM. You can see LSTM was very, very accurate because mean square error was kind of very, very small. but these are preliminary results. We have not finished on this one. And so preliminary conclusion can say BESPM has showed very robust performance compared to all other models because it was performing well and responding very well with the changes in climate. Thank you very much.
Oh, thank you. Questions? Questions? Oh, yeah, there are quite a number. I'm not sure whose hand went up first, but Lighton.
All right, thank you. I don't know if can you hear me? Hello? Can you hear me?
Yeah, we can hear you.
Oh, yeah, wonderful. Thank you. I had to confirm. Thank you for the wonderful presentation. Turns out we were working with our meteorological department not too long ago, doing something similar, although not really predicting yield here, but predicting rainfall patterns. I'm curious, the standard, right, when you're dealing with meteorological data is you look at the 30-year span. But if you go to the slide where you're showcasing the historical data you're working with, I think those were, unless if I'm mistaken, the historical data is over a period of 10 years or something. Are you able to justify the reliability of the results seeing as you're going against the norm? That's the first question. And then I thought it was really interesting that, so you mentioned that you're utilizing this already existing data set because you don't have a lot of weather stations. The obvious question one would ask is, did you try to look into the effect of actually using data coming in from weather stations? So did you identify specific areas within Tanzania where you have weather stations and compare the efficacy of the results or the effectiveness of the models relative to only using the Euro data? And then I was interested in finding out also if you looked into the effect of your models when you look at different or varying levels of detail, the geolocation. So district, I imagine the other boundaries that you look at, like the part of the world I come from, we have provinces, districts, we have wards. Did you look into this? And then finally, I thought it was really interesting when you showed us the attributes associated with weather data. What are the practical implications here? Like, what would it take to actually deploy what you just presented in a real world setting? If I'm a farmer and I wanted to deploy this, what would it take? Thank you.
Thank you very much.
Update one. Let's go.
Go
Let's
ahead
go
and
You
answer.
can respond, right?
Yeah, just respond.
Okay, thank you. For the first question on on 30 years versus 10 years, if you visit our lab, you found out we did a research on 30 years prediction using modeling techniques in 2013 to 2014 or 2016, where we looked on data from 1981 up to 2010. So one of the weakness of those, or one of the weakness of that system, we didn't have very good systems to collect yield data. So most of the data that we had so much errors that we cannot use. So we at least trust the data from 2005 up to today, at least a little bit. Still, we had to remove some of the district to make sure like our model does not react differently. And on weather stations, when you look at our website, there is one of our website that we are showing location of all weather stations across Tanzania. We know each location of weather stations, and we have modeled even the data from those weather stations. But these weather stations, the way they have been located is not that much strategic that you can use. But the good thing is with these weather stations and from TMA, they contribute data to Copernicus too. So those systems that use physics to try to harmonize the data or to harmonize weather and climate information, they use these weather stations to make sure like they're very good, accurate. And they have been tested by the climate experts in Tanzania. And this model has shown very good efficacy in places where there is no weather stations. So we at least trust that we can use this for prediction of yield. And another thing which is kind of very important you asked on wards in district. In Tanzania also we have that. We have we have regions, and inside region we have districts, And inside district, we have wards, and from the wards, we have villages. We have so many villages. We have more than 83,000 villages, I think, something like that. We have wards. We have almost, I think, 8,000 wards, and we have district 184. Now it's 184, I think, districts. One of the things that is kind of very important is the data that is being collected from the villages is being tallied at the ward, and it is reported as a village. It is reported in district level. So it's kind of complicated because they have a system they use, is called ARDS. So what they do is they do manual calculations from the village and ward. So the data we get from the village, you cannot even access, and they don't have even the records for it. So they have only district level records, and they have region level records. So it's kind of difficult to do ward level prediction. Another thing on how we can we can use this in real world settings. And here we're talking about very good inference engine. For the inference engine, so what we do with this one, if you load data, for example, if you load real-time data to this model, it can receive data. We'll look on how it can receive data from NOAA, and also how it can get data from TMA. We have discussed, not directly part of this project, but we have discussed on how we can utilize that. And because, if you want to predict for this season, you really need the data up to today so that you can predict for the next 6 months. Because for this model, the way we have trained, we have made sure like we only train for the half of the season and leave the other half of the season for prediction. So even for the prediction we are not using until to the last moment. Instead, we leave some part, or we don't use data or climate information for at least 6 weeks before harvesting. So in Tanzania, it's somehow technical, or we can say somehow tricky, because for real world prediction, we have regions that have what we call single season, and we have regions that have 2 seasons. So for this model, the way it has been trained to use for those double seasons, not for single season directly. So we cannot predict.
I think, sorry to interrupt you. I think you've given a very elaborate answer. We have maybe time for one more question. I see Shegun's hand up. I wanted to give him the chance. I hope if Lighton, there is still clarification, maybe we can do it on the chat. Kindly.
Okay.
Yes, of course, and I will reach out to him, Osami. Thank you.
Shegun, a quick question, please. We can't hear you, Shegun. Vanessa, do you have a question? I see you've unmuted.
Sorry, that's a mistake.
There are questions in the chat box that I may need to attend to.
Okay, so maybe Kadege, can please answer the questions on the chat, directly on the chat, to allow us to move on to the next presenter, Manasseh Matemba.
Okay, thank you.
Thank you very much. Okay, so if you're good to go, and then if you will indulge us, you can have your video on and we can see you as you present. You have 15 minutes.
All right. Good afternoon, Mr. Maina. Thank you for that. I'm just trying to share my screen. All right, thank you. Am I audible enough and can you see my screen?
Yes, loud and clear.
All right, thank you. Greetings to everyone once again. I'm Manasseh Samantha Matemba from Malawi, and I'll be presenting to you my progress report as a junior researcher. My study was investigating impacts of riverine flow regimes on aquatic biodiversity, a case study of Ruo River, an Afro-Montane freshwater ecosystem in Mulanje, Malawi. I'm a master's student at Malawi University of Science and Technology, studying biodiversity informatics. My supervisors for this study were Dr. Emmanuel Velemu and Mrs. Pokom Tewa. All right, so just to give a background on my research, basically water is an invaluable source and it can be very complex. Everything is connected to water and this gets to freshwater biodiversity. Freshwater biodiversity is important because of the benefits that come with it, from our basic needs such as food, food resources, water for domestic uses, and agricultural purposes. Freshwater also aids in climate regulation, recreation, purification, and nutrient cycling. And also freshwater biodiversity is beneficial to culture in almost every setting. So why is this study important? The study of macroinvertebrate aquatic species in freshwater systems. We cannot fully conserve what we do not know and what we have. And there is scarcity of aquatic macroinvertebrate data. The question is, what are we conserving and why? What have we lost and how? Because we cannot protect something that we do not fully know or fully understand. There is reliance on most data from other researchers from other countries, such as Tanzania, South Africa, within Africa. But Malawi as a country, we do not have any reliance to different types of studies. And also practice of biological monitoring of ecosystems is not really embedded in water quality monitoring in Malawi. So one would get to ask, why macroinvertebrates? Macroinvertebrates are small organisms that live for most part of their life in freshwater. These include, you may know them as stoneflies, dragonflies, snails, mayflies. So these stay most of their life in rivers and streams. They play a crucial role in maintaining the health of the environment. How so? By regulating nutrient cycling, they aid in the breakdown of nutrients and they aid in the movement of nutrients. And also they impact as predators to vectors. And they are food resources to fish and other invertebrates. Mostly we hear studies on fish, aquatic species, and it's mainly on fish. But those fish have somewhere that they begin from. They have something that they rely on, and macroinvertebrates are one of those things. So they serve as indicators of water quality. A certain species presence or absence can indicate a certain change. It can be either pollution or events such as natural events, cyclones and whatnot. We've experienced such as those in Malawi. So a presence or absence of a certain species can be indicative of a problem or a cause. So why macroinvertebrates again? Because they are generally abundant and easy to sample, and they present little mobility and limited migration patterns. So the research questions were really, do macroinvertebrates vary across different sites along the Ruo River? And how do different sites affect macroinvertebrate diversity in the Ruo River? The main objective was to investigate the impact of riverine flow regimes, flow regime change of aquatic macroinvertebrates located in an Afro-Montane area to determine the flow rate of the Ruhr River across its gradient at different sections, to assess the effects of changes in the Ruhr River flow regime from macroinvertebrate abundance, richness, and distribution, and to also to develop a database of macroinvertebrates using Ruhr River data. To give an evaluation of why this research is important, this research builds on biodiversity conservation and management. How so? It feeds on biological monitoring of ecosystem using biological organisms. It feeds into the water resources management of different ecosystems. It can be a representative of Afro-montane ecosystem. And also it feeds on information of ecosystem health and resilience. It also helps in data-driven decision-making. We have policies and practices that have to be concluded with certain data. Why are we doing this? What's the data and what are we achieving? So this kind of baseline helps in regulating freshwater ecosystems and managing their conservation. And also this builds in capacity building and research excellence. People like us are on to studying such aquatic organisms, and this builds also curriculum in different aspects of research. Okay, so taking you to the methodology of my study, the Ruhr River is a freshwater Afro-montane ecosystem. It is located in Mulanje Mountain Forest Reserve. Mulanje Mountain is amongst the highest mountains in Africa, probably the second. And Ruhr River is a key biodiversity area because of past research of a screeching frog that's native to Ruhr River. And it's significant for its water catchment value, abstraction for hydropower, and large-scale irrigation for tea estates. And it is a large tributary to the Shire River, which feeds into the Zambezi Basin. So the sampling was divided into different streams in order to get a representation of the whole area from upper stream of the Ruhr River and middle stream as well as lower stream. So these categories were done using elevation of the areas. Data was sampled for a period of 6 months from July 2022 to December 2022. Macroinvertebrates were sampled in the field and also carried to the laboratory for further identification and classification. So the study was divided into field sampling, laboratory work, and computer-based, where, like I mentioned before, there was macroinvertebrate sampling, and also river flow was measured using the traditional float method. Macroinvertebrates were identified in the field most of the times, and where possible were carried to the lab. They were identified to family level. Computer-based methodology was basically from the literature review to data analysis, database creation, and thesis writing. So this is an exhibit of me in the field and my different teams. The research exercise per month took about 4 days. To visit the upper stream would require 2 days because the mountain is 2,055 meters above sea level. So I had the assistance of research forest guides and porters. Moving on to the results. So from the findings that I've so far and the data analysis that I've done and still completing, this is what I'm going to present. Data was stored in Excel and also analyzed in Excel and analyzed in R software. So this graph simply represents the river flow at the different sites and the family abundance of the macroinvertebrates. Generally, river flow was found to be higher at the upper stream and decreased at the middle stream, and took a kind of an increase at the lower stream. And also the corresponding family abundance, it was higher at the upper stream, decreased at the middle stream, and showed an increase at the lower stream. There were also physical chemical parameters that were measured per each sampling, per each site. It is important that I also mention that these macroinvertebrates were sampled at 3 different biotopes, that is the gravel, sand and mud, stone and sand, and vegetation. So from the physical chemical parameters across the sites, they varied across the sites. Although they varied, Poisson regression confirmed that the physical chemical parameters were significant in shaping the abundance and the occurrence of macroinvertebrates. However, dissolved oxygen and pH did not show any significant effect. Moving on to the spatial macroinvertebrate abundance. This graph shows the different sites and the macroinvertebrates that were found at the site. With analysis of variance, the relationship was investigated, and there is significant evidence that macroinvertebrate varied across the sites. And also from the median of this box plot, there isn't significant difference in the median values that were across all the sites, because none of the median falls outside the median range. However, there is some data that is shown outside the ranges, which shows significant differences from one site to the next. In this instance, from the upper stream box plot, it shows more outliers, which may indicate that more families were exclusively found only in the upper stream and not anywhere else like the middle stream or the lower stream. And also in the middle stream, there are few outliers showing that the middle stream had little or few families that were not found in the upper stream or lower stream. The Shannon index also confirmed that the diversity by site was significantly different. A non-metric dimensional scaling was also done to look at the relationship of macroinvertebrates with different sites. It showed species association spatially. Certain particular species were associated with different sites. As you can see at the upper stream, we have species such as the Telagonid and the Dixid, the Hydrophilidae. It shows that they were mainly associated with the upper stream. In some areas, there was high concentration of certain species, while in others, they were not. From the lower stream, we saw there's an increase in the type of families of macroinvertebrates. Excuse me. These patterns help understand changes in the ecosystem. And this also shows species specificity, which indicates that there are specific macro habitats that are favored by different types of macroinvertebrates depending on their needs. And moving on to the temporal macroinvertebrate abundance. Macroinvertebrates showed variances in their abundance by month and at different sites. From this graph, it shows that at lower stream, generally, there was low abundance across all the months. At middle stream, it showed a higher abundance at first. However, in September, there was a sharp decrease, which only took a constant kind of increase from November to December. However, upper stream shows a different result altogether, where the macroinvertebrate abundance was high in July and decreased between September and October, and then took an increase from October to December. To give an example of some of the species that showed interesting results, for the order Coleoptera, these are beetles, they showed an increase over time from July up until December. And also an analysis was done on the EPT group abundance. This is the Ephemerotera, Plecoptera, Trichoptera abundance. Why do we use such an abundance to explain the results? It estimates most families that have low tolerance to pollution. The EPT abundance varied across the 3 sampling sites. For example, Ephemeroptera decreased from upper stream to lower stream. And also Plecoptera was absent in all other streams. It was only found in the upper stream. So these results, they suggest that the composition and structure of macroinvertebrates can be greatly affected by river flow, as well as physical chemical parameters. And then at a deeper glance, when you look at the different anthropogenic activities taking place from the upper stream to the lower stream, they show changes, and those changes can in turn affect the different biotopes and the composition and structure of macroinvertebrates. It also confirms that natural flow provides suitable habitats and conditions for different macroinvertebrates to survive. For instance, at a certain sampling, there were no macroinvertebrates found at all at the middle stream. What activities were happening at the middle stream? There was abstraction of water for a hydropower that's just that discharges water to the downstream. And it was only one type of macroinvertebrates that was found, the Oligochaeta, which is a snail. This Oligochaeta is only conformed to places of stagnant pools of water. So because of the water that was being held for the hydropower, there was stagnant movement of water, and some macroinvertebrates could not survive there, and only the Oligochaeta was found there. This is just an example of some of the macroinvertebrates that showed specificity. Even the beetles, they increased over time, and with the different streams, beetles indicate habitat alteration. So the research really shows that macroinvertebrates differed spatially and temporally, and also with different river flows. How could this be applied and implicated in our natural real state? Understanding the relationships between the communities helps to create strategies that can be used in restoring ecosystems. For example, it helps to prioritize which areas need more management. From this study, we can conclude that the middle stream needs more attention because this is where there are many land use changes, from water abstraction for hydropower generation, from to water for irrigation and other agricultural activities. So it really gives a basis on what steps to take in improving ecosystems. And also, besides generating numbers and graphs, the data is important to understand the state and functioning of freshwater ecosystems, what changes are happening. For this data, there is really no comparison on how we can see if there are any changes, because there hasn't been any data before that can help to see the differences. What have we lost? What are we conserving? But it can help in future. And also the research conforms and speaks to research pillars of environmental sustainability in the National Research Agenda for Malawi, which feeds into the Sustainable Development Goals, especially number 6, which emphasizes clean water and sanitation, feeding into target 6.6, which emphasizes the protection and restoration of water-related ecosystems, including mountains and forests and wetlands. So how do macroinvertebrates and aquatic species come into this? They tell us the changes into the ecosystem. They confirm what changes, and they act as a traditional method of forecasting the changes. Back then, we know about indigenous knowledge that can tell you what changes are happening. This has to be embedded even in the now machine learning and stuff to see how best the conservation of biodiversity can progress. And it also supports sustainable goal number 13, climate action.
I'll ask that you wind up, please.
Okay, thank you. And from this study, I managed to attend conferences, the UNESCO Men and Biosphere Reserves Youth Forum, because I was working in a biosphere reserve. I also attended the research design course in Kenya, and I also presented an abstract at the second Biodiversity for Malawi 2063 on spatial and temporal patterns of macroinvertebrate abundance on implications for freshwater ecosystem monitoring. The next steps to my study is completing the thesis writing, database creation, and data publication on IPT, and possibly leading to GBIF, and also research paper publications. 2 manuscripts are in progress. I would like to acknowledge some of the literature that I've been using, and also my research team from the Forest Guides, research assistants, my 2 supervisors, Dr. Emmanuel Velemo and Mrs. Tokontewa, for their support. I would also want to acknowledge the huge support from Data Science Africa that made most part of this research a success, and JRS Biodiversity Foundation and the Malawi University of Science and Technology, and above all, God. Thank you for listening.
Yeah, thank you very much. Thank you so much. I think let's maybe 1 or 2 questions. We unfortunately not too much time for questions. I imagine there will be 1 or 2. Maybe I'll start. For this kind of data collection, did you need to like do a number of repeats, or because of the how difficult it is to get to the sampling sites, do you just have 1 sample per location?
All right. Thank you for the question. I think the repeats were necessary to see the differences in the river flow, because the main factor before other interconnected factors that I was interested was to see the changes at different river flows, and those flows could be seen maybe at different months.
Okay, now I'm also curious, since it seems like you didn't do a full 12 months, could the cycle, and this is, you may correct me because I have no expertise in this, the increase in the population of the beetles, could it be purely cyclical? And that if you were to conduct this over maybe a few years, that you just see a cyclical pattern?
I think to see if it's a cyclical pattern would also take into consideration the changes in the environment, because I would have loved to sample, especially in January and February, where we had high flows, and even after, where we had the cyclone Freddy here in Malawi, but the area was dangerous and not accessible. But I think there would be changes. We would not call them cyclical, but it will be depending on the different climatic conditions.
Okay, thank you. I don't know if there is anyone else with a question. I see some comments on the chat. We'll appreciate you. Thank you very much for that. I'll then ask, I believe it's Rosemary, to proceed with uploading our presentation and going on and presenting our work. Thank you very much, Manasseh.
Thank you.
Manasseh, oh, sorry, it's you. Okay, Rosemary.
Hello, everyone. I hope you can see my screen.
Yes, we can see your screen. If you're able to show your face as well, that would be great, but you can proceed. You have 15.
Maybe I'll show it at the end because my bandwidth is not that good. I'll show it at the end because the internet may flicker on video. My name is Rosemary Nalwanga, and I'm grateful for this opportunity to present the work I've been involved in, entitled An Edge AI-based Smart Irrigation System, with my supervisor, Dr. Inyas Katayo from the University of Rwanda. With our work, there's advancement in technology, and this has led to growing popularity of precision agriculture. In Africa and the rest of the world, population basically depends on agriculture for food and also factory products, industrial products, and it makes a high demand for agricultural products. And for us to have this demand serviced or satisfied, there has to be a lot of technology put in the agricultural sectors to enhance production. And so emerging areas in Internet of Things and AI have been used to enhance agriculture, basically precision agriculture. And that is why for this project or research, we were looking at how we can involve Internet of Things and AI to be able to boost on production based on one sector of agriculture that deals with irrigation. There are very many benefits, such as optimization of resources when we take this trend of AI and IoT, and also helping us to take informed decisions in all the operations that are being taken, leading to the food production. In the problem statement, because of the increasing demand in agricultural products, there is need for precision farming systems that can help farmers to be able to take informed decisions on all the operations they are making, so that they can have a high yield and production at the end of every season. Smart farming is used in attempts to increase the production. And looking at the irrigation, we have very many places that are not good with water in the soil, which is soil moisture. And also because of the climatic changes that keep happening every now and then, sometimes farmers plant their crops and then rain does not show up. So irrigation comes in as one of the things that can be used to enhance the crops so as to grow to full maturity. However, there are a number of solutions that exist that some are manual, some others are automated. Like we know that with automated systems or technology, we so much look at threshold values, which may not be very accurate with our changing climate every now and then. So the problems that the current irrigation system have, one, there is water wastage, because when those pumps are opened, they can irrigate until the farm is even flooded somehow, in case it is not being monitored at no time. And then also most of them are manually operated, and because of the wastages and the manual operation, of course, it comes in with an increased cost of operation. So the main objective with our research is designing a prototype for an edge AI-driven irrigation system. With edge AI, we look at developing tiny machine learning models that have that require very less memory and computation power to do the processing. And because we know that most of our edge devices are constrained in terms of memory and storage and computation power, we require models that are smaller to fit on those devices. And also in Africa, most of our agriculture activities happen in rural areas where we do not have connectivity and infrastructure to be able to access the cloud. And so with edge, deploying these models at the edge comes in a way that it helps the farmers to be able to utilize this technology even without the other infrastructure, and also reduces on the cost, for example, for the bandwidth that may be required to send to the cloud, do the processing, and then send back to the device. So the specific objectives that we followed with our research, one was exploring the hardware and software requirements for implementing a smart irrigation system, and then developing algorithms and models to determine the optimal irrigation timings using AI techniques. Number 3 was to design a user-friendly interface, allowing farmers to monitor and configure the smart irrigation system depending on their location and the needs in those various locations. And lastly is to prototype an AI-driven irrigation system for an African setting. With African setting, it looks at what I just explained about the infrastructure and connectivity lacking. So in the African setting, we have to be considering that, and also considering what farmers in the rural areas can be able to access. And some of the identified gaps in the current solutions, as earlier discussed, the issues with the manual-based systems that also come in with the cost, and also online-based solutions are not appropriate for Africa because of the connectivity issues and then infrastructure. And also solutions do not consider forecasted weather data that and plant water needs and the plant water needs. With the forecasted weather in Africa today, there's research ongoing focusing on so much of weather, because weather keeps changing. We no longer have seasons like we used to have them. They keep changing. You cannot predict that. You cannot just think that maybe this month we shall have rain. Anything can happen. So with that solution, we want to look at how we can also incorporate this forecasted weather, so that in case we are sure it's going to rain, we don't have again to waste with the irrigation system that is installed on the farm. And what we thought about something, why would we use AI other than any current smart systems? And we had basically 3 reasons. One is the water efficiency. According to a study that has been published, when we use AI-driven irrigation, if we can use AI-driven irrigation systems, we can reduce water consumption up to 40%. Also, energy efficiency, reducing on the energy consumption by 30% compared to the energy that would be consumed using the traditional methods of smart irrigation, and then also crop yield optimization. With the work, the methodology that we took, we used a number of tools, and the central tool that we used here was Edge Impulse, and this one we used it to be able to develop a tiny machine learning model that we deployed on our Nano PL Essence. It's a microcontroller that has Cortex that can be able to accommodate that tiny machine learning model. And then with that Edge Impulse, we used TensorFlow for the machine learning, for the deep learning or machine learning or training of the data. And then Arduino, because we are using the microcontroller as, which is Arduino Nano. And then ThingSpeak cloud platform for that we've been using to be able to gather the data that has been collected. Because another challenge that we face is accessibility to data, because with machine learning, data is something very important. When you're training models, you need data. You need at least enough data to avoid the issues of overfitting, to avoid issues of exploding gradients and vanishing gradients. So anytime there is an opportunity of gathering data from sensors, from fields, and then it can be kept somewhere probably for further use, it comes in handy. So that's why we had to use also ThingSpeak. And with our proposed system design, we have the microcontroller, which is the Arduino Nano PL Essence, connected to a power supply. One of the methods that we intend to use when the system is fully deployed in the farm is using solar energy that can also help to maintain a green environment. Having a forecasted weather API that can be pulled maybe on a weekly basis to be able to help know if, how the weather is going to be. And then our major sensor was the soil moisture sensor, plugged in the soil and then sending data about soil moisture to know when to irrigate and then when not to irrigate. And the temperature and humidity sensor also was used. However, in the process, we realized that soil moisture sensor can as well give good results without using the temperature and humidity sensor for purposes of reducing on the cost, because again, we do not want the farmers to incur extra costs, operational costs because of the system. If we can, if there's a way we can avoid that, it would be good. So we felt like in the final system, if implemented and on the field, we could just rely on a soil moisture sensor. And then having a GSM module that can send a notification maybe to the farmer's phone on what is going on, and then a relay connected to the microcontroller, which is the actuator. When there is need to be irrigation, then it can send a signal for the water pump to be turned on, and then an LED light emitting diode for status to know whether the system is on or off, whether it's irrigating or not. And then some of the hardware components that we are having on our system, we have the Arduino Nano, we have the water pump, and then a relay, and the soil moisture sensor, and the GSM module. With the data collection, because we needed data for us to develop our model, the TinyML model, we collected data for a period of 6 months from a farm, and also we tried comparing this data to data that is online to see if there would be a big discrepancy in the data that we are using. And then we realized the data that the difference was small, and that at least gave an assurance that the model is kind of accurate, depending on what we wanted to predict. And when we tried testing the model, we had an accuracy when it's for irrigation, the accuracy is 96%, and then for no irrigation, it was 88.8%. So the F1 score is a metric that is used to measure model accuracy. It gave us 0.94 under irrigation, and then under non-irrigation gave us 0.86. We tried testing, of course, from the lab, not from the field, because like how you can see our prototype, there's wiring, breadboard, it would be not feasible putting in the field, but we got like a soil sample and then plugged in a moisture sensor, soil moisture sensor, to be able to monitor what is going on. And all the components were connected together, and our deep learning model had been put on the Arduino Nano BL Essence, and then we monitor and see what happens. The water pump is also connected to know when to irrigate and when not to irrigate. And of course, our sensor was also calibrated so as to avoid values that are not within the range of the field that we were considering. When we tried to do a sample prediction output, we realized that the model was giving us accurate results, both on the device, and we also tried testing to see on the cloud if the prediction would still be the same, and the results were, the prediction was still the same, and also the F1 score was close to each other, both on the cloud and then on the edge. And that was just purposes of testing to see what would happen if we would get the same results that we would get at the edge and the cloud also. So in conclusion, implementation of this system will help solve the existing problems and reduce also on the wastage of water in relation to irrigation and smart farming, also leading to improved production and reduced costs, because when we look at a fully implemented system, we feel it can be affordable to the farmers, even deep down in remote areas. And the system also contributes towards the achievement of the Sustainable Development Goals, the goal of no poverty when we try to minimize on costs. Improved production also will ensure farmers have enough to sell and to eat, reducing on hunger. And lastly, responsible consumption and production. Thank you very much. Thanks for listening.
Thank you very much for the presentation. We welcome questions. Maybe my first question would be, You showed us the prototype in the field. This last picture you showed where you had a solar panel, did that mean that you moved from the lab, you were able to build a prototype that you could deploy in the field?
No. That prototype that I showed in the field was in one of the fields where we went to see, to collect some data, because they have a farm that it is based on automated system. So they have that deployed. So that was in the field. It was not my deployment. Ours was in the lab. We haven't like deployed our prototype in the field because it is still on breadboard, like how you saw. So that one was like an illustration of one of the farms that we went to. It's not our deployment.
Okay, other questions?
Hello, everyone.
Hello.
Yeah, this is Alcado from SUA, Sokoine University of Agriculture, Tanzania. Thank you, Rosemary, for your interesting presentation. I have actually like 2-3 questions. One, from the objectives, that you are designing a user-friendly interface which will allow farmers to monitor and configure the smart irrigation system. So I would like to know where the interface is. This smart, smartphone based app that should be providing this user-friendly interface to farmers, where they can like click or configure or automate their irrigation scheme, or you see on the device in the farm, where they will be like visiting the farm, click maybe somewhere, or press a button to irrigate. That's 1. 2, what do you mean by an edge AI-driven irrigation system? Do you use any edge computing technology or facilities? How do you implement that? The last one, which AI techniques do you use in your implementation? Thank you very much.
Thank you. I'll start with the one for edge, where you said what we mean by edge AI-driven smart irrigation system. With artificial intelligence, where we started with concerning artificial intelligence, most of the data collection, data processing, storage has always been done at the cloud. Yeah. So with Edge AI, we look at how we can have data processing, collection, computation, and storage, everything at the edge. With edge, we mean that devices being at the edge of the network. For instance, in our case, we have a microcontroller, and then we have a sensor, and then we have an actuator. So we have our model that is deployed on the microcontroller. So we don't have to send data to the cloud and then processing is done. And so we can have inference at the device. We look at having everything on the device, and that's why we call it edge. So it is edge AI because the artificial intelligence or the inference is being worked on at the device itself without having to go through bandwidth and latency issues of sending to the cloud and then back. So that's why we call it edge AI driven. It is our AI deployed on an edge device and then helping with the smart irrigation. And then the first question was, can you kindly remind me? Okay, let me first answer the last one, then you remind me the first one. For the last question, you asked the techniques that we're using with AI. We are basically using deep learning, and this we are utilizing a platform which is called Edge Impulse. With Edge Impulse, it helps you to go through all the steps of creating an impulse, which is like a model. So we do data gathering, we gather data, and then we upload it. That data has to be prepared well with its labels. So when you upload it on the platform, you can use a pre-trained model to be able to train your model until it learns to be able to give you inference. Then at the end of the day, you get that model and then you can deploy it on your device and be able to do the prediction or the inference that you need to do. And kindly remind me of the first question, because I think I've forgotten.
Yeah, thank you. The first question I asked that you are developing a user-friendly interface which allow farmers to monitor and configure the smart irrigation system. So I'd just like to know how is that designed? Is it a smartphone app that will allow users to like monitor their farm or click somewhere in the app to automate the irrigation system when, for example, the water level drops down or goes up? Or is it an interface that is probably designed on the device which is located in the farm, where a user needs to visit maybe his or her farm and press a button, automate the irrigation system? How is that user-friendly interface designed?
Thank you very much. So we have two ways of communication to the farmer. One way is using a mobile phone. That is, we have a mobile app in place that we are still testing whether it can really send on time. Like you saw on our prototype, we have a GSM module. And so it can send a notification on the farmer's phone. Yeah. However, even on the device itself, there is an LCD for purposes of prototype, like an LCD that a farmer can look at and know what is going on. But the system itself, we do not expect the farmer to be part of the process, like operating as such. They just need to be monitoring and knowing what is going on. Otherwise, one of the reasons we talked about is trying not to go away from the manually operated irrigation system. So the farmer doesn't have to go and click a button. The actuator is also connected so that if irrigation is needed, it triggers the water pump and then it irrigates. And then at some point, it is supposed to trigger it to also stop, so as to avoid wastage, because the moisture sensor keeps in the soil. So when it realizes that, it senses that the moisture is what is required, then also the irrigation is supposed to stop. So the farmer does not require to press a button. Also powering the device, for implementation, we intend to use solar power, solar energy. So the farmer can always visit maybe the farm to see what is going on or monitor from their mobile phone.
Okay, I think I'll jump in because we are running out of time. If there are other questions, kindly put them in the chat. I think you've answered Alcado quite a bit. I want to invite Samson. And thank you very much, Rosemary. Thank you for the presentation.
Thank you.
We give you a clap.
Thank you.
We look forward to you deploying in the field.
All right.
Okay, so I believe Samson, we can hear from you.
Yes, I'm ready. Let me just upload the slides. So good afternoon, everyone.
Good afternoon.
You can see the slides?
Yeah, but they're not in presentation mode. They're in edit.
Now they should be. Okay.
No, I think it's still, we see, no, we see quite a number of slides. I don't know what mode this is, but
okay, let me see. Okay. Let me just reshare so that it can.
You may just press F4 key. You may just press F4 key.
It's clear now?
I think it's fine now.
Okay, thank you. Yes, so I'm Samson Oko, and the presentation is on TinyML based self-diagnostic kit for respiratory diseases. And this we are doing together with Dr. Jimmy Nsenga of the University of Rwanda. And I want to move to the next slide. Okay. On the motivation, basically, increasing deaths and disabilities. In fact, data from the World Health Organization say that almost over 4 million people die yearly because of some of these respiratory diseases. So we thought of tried to find out what is the reason as to why the numbers are high like this. And one of the main reasons is that most of the times when people have this respiratory disease, they notice or they go to the hospital when they are severe, or when some of the conditions have gone very far. So we thought of what if somebody has just a device at home and you can be able to, maybe in the morning before you go to work, try and check your health status as you go. And it makes it easy to limit some of these cases so that you can be able to detect some of this early. And when you look also at the diagnosis of such diseases, you find that in Africa, we have a challenge of medical experts. So, and also the devices that you need in the lab to be able to take some of the tests to come up with this. So we looked at the use of some of the latest technologies, basically IoT and machine learning, to be able to apply in such cases to help us to be able to come up with something that can help us to try and know if somebody has a respiratory disease or not. And then another challenge is data set. That is a problem in Africa. And so with this device, if you have so many people in different places with these such devices, they can also help us to get lots of data that we can also use to be able to do even future research and to help in other areas. So the aim basically was to enable a day-to-day chip-free respiratory disease self-diagnostic kit for the African population, and basically by relying on IoT and AI technologies to be able to come up with a rapid kit for respiratory diseases. So the first thing was to understand how respiratory diseases can be detected from exhaled breath, and then identify some of the IoT and embedded technologies that we could use to be able to transmit exhaled breath signatures, and also to identify some of the open source AI and non-AI technologies that we can use to detect respiratory diseases, and to design a prototype that we could be able to use to be able to diagnose respiratory diseases. So as we were going on with this, some of the technical, I can call them technical research challenges that we had. First was breath collection, designing of the breath collection tube and the sensing unit. You have to ensure that there is no interference from other outside the environment that you want to collect the data from, and you can't put a sensor into somebody's mouth, so you have to come up with a design of where somebody blows into the air, and then you're able to collect that. And then also, in terms of sensing, no commercial sensors for breath VOC, so we try to explore the use of the commercial gas sensors that have been, at least from the manufacturers, information can be used for detecting some of these breath VOCs, and also the problem of data sets to be able to train the AI algorithms. So that was also another challenge that we had. And also how to be able to move this and to be able to detect on the device. Because if you're going to apply machine learning and you want to have a self-diagnostic kit that somebody is using at home, then you would want something that I don't need any connectivity or something to be able to detect that. So we were looking at moving now my model to the device so that the users are able to use that from home. And when we looked at what has been done and the basis for integrating IoT, machine learning, and edge computing, basically, to be able to predict respiratory diseases, is that there is what we call breathomics. And from studies, it is shown that the exhaled breath that we breathe out, so when you have some diseases, respiratory disease, the components, the volatile organic compounds in that is not the same as somebody who is healthy. So there is usually a difference. And so we thought this is an area that we could explore to be able to see if we can use that. And then breath samples can be directly sampled into an analytical hardware. And that was done by one of that. And then commercial gas sensors also been shown that can be used to be able to collect exhaled breath data. And so we went to explore such. And also the use of deep learning models are basically becoming popular, especially when you're dealing with larger data sets, to be able to predict some of these conditions that people have. So in the state of the art, we try to see what commercial solutions are there, and there are some that they call spiral nodes that integrates electronic nose technology and spirometry. There is TrialSmart, that is a breath test commercial solution, and breath track analyzer to guide doctors on diagnosis. And there are also some open source prototypes that people have tried to come up with that are able to, you can be able to use to be able to detect some of these breath volatile organic compounds. However, the commercial solutions, we noticed they were very expensive, and also they were dependent on cloud technologies. So that means that most of the times somebody you have to be connected to a cloud platform, then the data is sent there, and then from there they're able to do that. And also there is also the lack of data sets that you could be able to use. And as we know about data sets, especially when it comes to machine learning, different areas, different people. In fact, there are studies that have shown that the data set that you get from an African is not the same as that you get from somebody who is from a different part of the world. So that is also something that we thought we could be able to look at. So some of the challenges with this cloud-based artificial intelligence technologies are basically, the first one that we found was connectivity, where being able to enhance portability to areas that have connectivity challenges. Then we have privacy concerns. People don't want their health information or information about them to be able to go over networks. You are not sure who is getting the data and that. And then there's energy, being able to, given that some of these embedded devices are energy constrained and they have resources that are constrained. So we want not to be able to have them on for long, and also costs that will go with that. And interestingly, when you use TinyML, you now move the AI model from the data source to where the model is. So unlike the cloud-centric, where you move data to the cloud, then the model is done, and then you send back the prediction. With this, you are moving the model to the data source, and then you're able to have your prediction at that. So for the system development methodology, basically we did prototyping, and first we had to come up with the functional and non-functional requirements. And then from the non-functional and functional requirements, we came with what we call the design constraints that we wanted to have on the device, and then design the prototype, and then testing, and also trying to deploy it and use it to be able to collect data and see what we are able to get. So for the machine learning, the training was done in the cloud, but once after training, we have used TensorFlow Lite to come up with a small model, and that we are able to deploy now on the embedded devices at the edge to be able to predict. So for our TinyML tool stack, we have the data, that is the breath VOC data, and then we apply deep learning to be able to do that. Then from that, we are able to compile and debug, and then on the embedded IDE, test that, and then come up with the executable that we were able to deploy on the device. We also tested this on Proteus to be able to see how it's performing in a virtual environment before we are able to deploy to the devices. So for the system, we have the Arduino Nano 33BL license, and then we have the SD card for collecting the data, and then we have the LDED and OLED to be able to give notifications. So if some health problems have been detected, it's green, and if it's not, it's red. And also the ESP Wi-Fi module, basically we were using it to be able to try and see how we can be able to send the data to the cloud, especially when we are training. And also we have got a rechargeable battery to be able to power the device. So for the gases that are targeted, that were total, there were 4 gas sensors that target the total volatile organic compounds, the formaldehyde, carbon monoxide, and air quality of the exhaled breath. And then data was collected from 60 healthy people and 20 people who had respiratory diseases. And from that, we are able to train a model. And at the input layer, we had 28 features, and with 2 hidden layers of 20 neurons and 10 neurons, and 2 output layers that shows whether somebody is healthy or not. And from the confusion matrix, the healthy was at 96.7% and unhealthy at 94.1%. And we tried to also do inference at different places using different platforms. On Proteus, we were able to virtually test this, and it was able to show the results. We also had it on a local browser, that is on a local device, to be able to see if you can be able to predict that. We also did inference on the embedded system. And also we had a Node.js application just to see that the result was according to from different aspects. And when you look at the real-time embedded process specifications, you can see that the model is tiny enough. Like, for example, in the STM32 cube, we are using 19.53% of the process, and of the RAM and the flash of 29.44%. So the model is pretty tiny enough to be able to run on some of these edge devices that you can be able to do. And also on optimization of the model. So if you use a Float32, an optimized model, you see you require more resources, but if you optimize the model, then it gives you at least better results to be able to implement and be able to do that on embedded devices. So, and also the data size. So as you get more data, we were able to see at what point we are able to get enough data to be able to train the model and try to plot the accuracy against the data sets to see how the model was performing. And some of the things that we've been able to do is that we have had one paper accepted, and I think it will be presented in November. We had one poster presentation and at least a technical workshop where we have been able to talk about the use of TinyML. And I want to thank Data Science Africa because of the devices that we are able to get. There are other people who are doing other research in different areas, but we were able to bring them into TinyML, and they've also gained from our devices to be able to work on their research and to be able to prove the use of TinyML in a wide area or in a wide range of application areas. And I think this is some area that we are still exploring and we want to improve more. We are at the point of wanting now to have the device that the way we have been using it's not portable. We have still been having cables all over and collecting data with that, but we want to package it, and we're working with some of these embedded device assemblers to be able to come up with a device that we can be able to send or give somebody and they easily move around with and to different areas and even to the homes to be able to use it. I think that's all, and I want to thank you very much, and I think I can welcome questions.
Thank you very much. Right on time. So questions? Questions for Samson?
Yeah. Oh, sorry. Okay, I'm there. Yes.
Yep. Do I see questions? One question I would have is what diseases actually manifest in terms of the sort of, I don't know whether the term you'd use is the compounds that appear in the breath.
Mm-hmm.
So which diseases are known to make that composition change?
No, basically, if at any time, if as a person, when you are not well, the composition changes, especially if it is affecting your respiratory tract. So, or at least I can say all diseases will affect that composition.
I see, and this is something that's medically well known and proven.
Yeah, that is well known and proven. Now, what we are trying to do that I think we have not been able to narrow down to is to say that this person has COVID, this one has asthma, this one has this. So that is some of the tests that when we get more data, we can be able to train and see if we can be able to differentiate.
Ah, okay. Thanks for that answer.
Yeah.
Okay, do we have more questions? More questions? Any more questions? Okay, if not, what we'll do, let's appreciate Samson. Thank you very much for that presentation.
Thank you, sir. Thank you.
Okay, I believe right now it should be Boniface. And I see you ready to present.
Yeah, so let me just share my screen.
You can go ahead
The
and
screen.
share, and you have 15 minutes.
Sure, can you see my screen now? Hello?
Yes, I don't know if I'm seeing
perfect.
a screen or, okay, but I see a screen, yes.
Perfect. Perfect. So the title of my presentation is Unlocking the Medical Data for AI Health Research in Africa using GeneTalk as an example. So first, who I am. I'm a master's student I'm currently at Strathmore doing data science and analytics. I'm also a GeneTalk active contributor and a huge free software advocate and enthusiast, currently contributing to upstream gigs and also maintaining gigs bioinformatics. And my supervisors are Piotr, Solomon, and George. It's nice that they are currently here. So a bit of introduction about my work. So one of the biggest challenges that's also we've seen in this presentation for the past 3 weeks is fair data. You know, having to find data that's findable, accessible, interoperable, reusable, and not just between human beings, also to machine agents. Being able to have access to data or like provide algorithms, machine learning algorithms data without any human or little human input. How do we go about that? And creating or making your data fair helps in unblocking or like helps in making data more accessible to machines and also more easily shareable between different organizations, thereby like helping unlock scientific research and also just making that process faster. So a bit of background for GeneTalk. It's a long-running biomedical web service. It's accessible through, it's currently accessible through our web interface and APIs. And currently it focuses on genetics data, phenotyping, detail, and genome-wide association studies. And a cool fun fact about GeneTalk is it's one of the oldest databases in the world, at least for genetic and genomic studies, since it has been around since '94. So what is the motivation, like why? And the title highlights what at least this project is trying to do. One, unlocking biomedical data. That means making data fair. And why is it important for that data to be fair? So that we can use it in actual AI health research in Africa. And one simple example of that is inputting data to like large language models, and I'll touch on that later. And finally, using GeneTalk as an example, I want to demonstrate or like showcase this project's approach to how we are able to achieve making GeneTalk data fair. So how? One, we actually convert the tables. GeneTalk currently has most of its data is in SQL format. And we've been using SQL since the 90s to date. And the problem with SQL is it's a very rigid 2D structure. And I want to show you how we're able to convert that structure from SQL to a graph RDF in a reproducible way, to a flexible way, and also a way that other labs out there, even outside labs, even other industries can also just adopt. So an example. So before I begin into the details of how I go about it, like what's this IDF I'm talking about and how does it look like? So I'm going to just highlight an example of how IDF looks like. And in this case, the example I'm about to show you is how we're able to use IDF to model data using an ontology I came up with, a classification ontology I came up with. In this case, we see we have a term called species, which is a classification. It's a classification level as part of some tree with a depth of 3, and it has the following members. And not much later, below the SNP, you can see that I classify strains using this classification stream, and also add extra annotation data to the strains. So just from this very brief snip, you can begin to see ideas of how the fairness of this data is and how well annotated it is compared to what a lot of people are used to. So the next question becomes, how do you come up with the above starting from SQL? So the most flexible way is, or the biggest contribution, at least for this project, was creating a declarative domain-specific language that can pass SQL to RDF. And what are the features of the thing I'm about to show you? One, it's database agnostic. What this means is I can give you the programming language and you can use it on whatever SQL database you have. I don't have to know about it. I haven't had coded it against any of GNetwork's database. The second really cool thing is, at least when creating the syntax for this language, I need it auto-documenting, which means as you're passing the data, it gives examples, and it also tries to explain some of the things trying to explain how it does some of the things. I'll show an example later. Another really nice feature is, since it's written in Guy, which is a Lisp, it means that you can use the full feature set of scheme, right, just within the DSL to do whatever it is you want to do. Then the last two, you have very flexible queries. Anything you can do, or like most of the things you can do in SQL, you can do in this DSL. Then finally, you have optional dumping of table metadata, which means that as you're dumping your data, you have a table called A, you can dump data about the columns, the data types of the columns, and whatnot. So that's metadata about the table. So let's head into the syntax of how this thing looks like. Before I get into the details, it has been inspired by two things: functional programming and declarative programming. Functional meaning that you have pure functions. For every input, you get an output, and declarative, you say what you want, you define what you want, you don't say how you're going to do it. So what does that look like? So here's an example. You can clearly see that we have the function, the syntax with documentation. The name of the thing is called GenBank. You have connection parameters. In our case, we don't want to dump table metadata, so we say it's false. Now here we have one of our high-level inputs, the prefixes that's going to wind up at the end of our RDF store. At the end of our RDF output. Then we have a list of inputs. In this case, it's a definition of a GenBank, which is a transformer. And then we clearly specify the outputs of where we want the results of our transformation to go to. In our case, it's a markdown file and a graph file, a TTL file. And when defining one of our inputs, GenBank, this is how it's done. And for this, I'm going to walk you through how it works by creating my screen. So on the top screen, you can see that we have a defined transformer bank highlighted here. We use the table GenBank, which is left joined to species using species ID. It dumps schema triples. This section allows you to extend your own annotations, and you're flexible to do whatever you want. And I'm going to go on this lower screen and show you how that looks like. One, you can see that this GNC nucleotide gets dumped into these triples. So you can use the schema triples to really define extra ontologies you have in mind. And then for each element of your SQL table, or rather for each row of your SQL table, you have a dump. In this case, we see that the subject is a GenBank, which has a sequence and it's classified under something. And you can see that here we have a GenBank, which has a sequence and it's classified under this species. Yeah, and that's our Turtle format. So what does our auto-generated documentation look like? For that, I'm going to head to our browser and show you an example of a Markdown. So here's an example of like the auto-generated docs just from our syntax and how we define things of our species metadata and the classification scheme we have. We have an example of how the exact SQL query that was constructed, an example of how the query results, or like how the query results or maps to triples, an example SPARQL query that you can run on a running graph server, and then an example of what to expect, in this case, expected results. And for this particular generated example, we had several inputs to that, to our syntax. So we have several examples highlighted. So back to our slide. Another big deal that I was able to do as part of this project was host GNetwork metadata online for other people to use, right? And I'm going to show a very brief example. So this is the link to the GNetwork Virtuoso server, right? And this is an example of how to query any of our metadata. And you can see here that you're really flexible to choose whatever format you want. In this case, I can choose a table. And here, I want to describe a resource that is specialized under a GNC set. So this is just like a live example. And here we have our results here. We have species, we have a query that is able to describe something that we did not know beforehand. And this very simple example demonstrates or like highlights how a machine agent can go about discovering data on its own and mining data on its own accord, as long as how you provide that data is in a way it can understand. And of course, if a machine can do a good job at it, it also implies that also as humans, we can also be beneficial to how we fetch data. And you can think about a common problem in Nairobi where you have different facilities like hospitals where they want to share data, but they can't because they don't have one unifying format. And a hacking solution many people have is to expose, say, RESTful endpoints, right? Then what happens if something changes? It's complicated. So the gist of this entire project, as you can see from the results set here, is to provide a language where you can transform your data into a format that's fair, into a format that has been proven before to make data sharing easier and also to make ingestion to machines easier. So back to the slides. I also have like the public repository listed here. Then I'm going to summarize my achievements, some of the challenges, and some of the remaining work I want to do. So in terms of achievements, it was a big deal that I was able to create an auto-documenting DSL for passing SQL to RDF. I've just demonstrated how I converted GNetwork metadata to RDF and exposed it on the internet for people to have access to. Another really cool thing that has come of this work is that the generated RDF is currently being used as an input for a large language model that GNetwork is working on. In terms of challenges, the biggest I had was passing very big data sets. But that's something I can work around. The priority for now being able to have something more like a proof of concept before like finessing everything else. In terms of remaining work, the one thing I really need to do is host the current work I have in Kenya and also extend some of GNetwork's data with local data. And that's with some liaising with the Cambridge guys. Another thing is assessing the ethical aspects of access to human data in the Kenyan context, given our privacy laws. And then the final one is publishing a paper. So on regards to publishing a paper, we have two current papers we are working on. One is documenting at least the DSL I created and targeting it to bioactive, and a related paper is on the LLM stuff. In terms of future work, I'm really keen on creating a more intuitive prologue-like DSL for querying RDF, because I feel SparkQL, the query language for querying Virtuoso like an RDF store is a bit hacky. So it would be nice having a more intuitive prologue-like domain-specific language to handle that. And another one that I'm considering adding is extending it to other data formats, JSON, CSV, and maybe other database formats. So that's it for my presentation.
Thank you very much. Again, yeah, right on time. So yeah, well timed. Very interesting presentation. Questions? Questions for Boniface? Oh,
yeah, Shelby. Hi, Shelby.
Go ahead and unmute and.
Okay. Oh shucks. Okay. Can you hear
I
me?
think you're fine. You're just seeing yourself on Boniface's
Okay.
screen.
Yeah, I was seeing. Yeah, I was seeing myself on Boniface's screen. So, I'm one of his advisors. Where I'm working on the generative AI software for our system, Gene Network, and one of the reasons there are several reasons Boniface's work, Munyoki's work, is so exciting and important. So he's using Guile and Lisp, that Lisp-like language. Lisp is the original language that was used for artificial intelligence development. And one of the cornerstones or foundations for doing any type of artificial intelligence work is data retrieval and representation, as well as management of that same information. And so, all the information that you're able to query using Munyoki's system, using being able to use RDF and having these fair and open formats goes just perfectly to the National Institutes of Health research that we do at UTHSC. And as I'm working on this LLM, it's going to be an expert system to support researchers, people who are specialists and people who are just interested in the type of genomic research that we do. It's going to be part of an expert system that we're building over at the Genetics, Genomics, and Informatics Research Lab in Memphis, Tennessee. So it's very exciting. We're happy with this progress, and there's so many other things lined up for him. So yeah. Thank you for letting me comment.
Yeah, thank you. And it's good to see the supervisors on board. It's interesting that you bring up this issue of expert systems because that's probably the origin of AI. And then now we've moved into all these other things. Any other questions? All right. I think I was curious. Who exactly you feel would be using the system? Is it designed to be used by what level of technical proficiency? Are they the biologists or people who generate the biological data, people developing models, all the above?
That's a great question. So the idea is it's targeted for everyone, right? Because it's a domain-specific language. It means that I get to choose the syntax of how everything is defined. So the idea is anyone who hosts data, think of it this way, right? Like you work in a university, I work in a university, there's someone else who works in a university, and somehow all the DBAs, the database administrators, need to find a way of sharing data across universities without having to hard code APIs and whatnot. How do they go about that? It's more work when they come in and they try to write the actual hard-coded language to get and glue things together. And the goal of this DSL is to provide it to people so that it's an easier way of passing something to something else. All they have to know is a very small subset of a language. Yeah, so it's really targeted for everyone, but at least for my current work, I'm doing it with the biologists because we're prototyping stuff and also we're showcasing its use case to like the rest of the world. Yeah.
And
So,
how receptive are biologists to doing this type of coding, or whatever minimal subset of the language they need to know?
Surprisingly, really receptive, and at least my experiences here has been because of the pain they have to like endure to get data from different labs far outweighs the pain of learning something new that they're uncomfortable with. So someone would rather learn something small, right, so that they can be able to do something rather than go wait in a lab for 5 years or like 5 years, like a couple of months or like weeks to just get access to a certain type of data. In fact, at least in our team, there are some biologists who are like championing the acceleration of migrating most of our work to like the GraphDB so that they don't have to go straight to the DBA to ask for data.
Okay.
Yeah.
Thanks.
That makes sense.
Yeah, it does. Any other comments, questions? Boniface is our last speaker for this series, so we can. Okay, Shelby,
may
you
I
can
speak
go again.
Okay,
Yeah,
thank you.
sure.
Thank you so much. So, what happens is everything Munyoki is doing is looking very complicated and very technical, and because there are some biologists, some of these researchers, they will learn how to use it just because they want to be able to access data well, but we're also making a more simple interface where they can use plain language to interact with it because since we have these large language models that work so well, why not use them as much as possible and to make the difficult things plain.
Interesting, very nice. So maybe we'll be hearing about that next time from Boniface. Okay.
Yeah, sure.
All right. So I think that brings us to, maybe I'll just go back on. So it brings us again to the end of this sequence over the month of September. And what will happen is that we'll be contacting you again at the end of the period for which you're doing your projects to give us a new set, an update really. So we are really happy with the way things have gone. We hope everything is going well for you. I think we request that you upload whatever evaluation forms we've posted to you so that it will allow us to then go with the other process of review and disbursement of other future tranches. So unless there is any comments, we can actually close at this point. We can first give Boniface a clap, and then unless if there are comments, feel free to just unmute and speak, and then we can close the session. Boniface, I think you can stop sharing your screen, and then it'll. Okay. Anything from anyone? Dina, is there anything I've forgotten?
I think that's it, Shira. They've done really remarkable work, and we really look forward to reviewing the presentations and share. And we really look forward for the open access to your data set once the project is over. But it's very good progress from everyone, and we're really excited. Yeah.
Yep. So did I see, Shelby, did you want to say something?
I was just gonna do what my colleague did. He sent a chat and said thank you for this initiative, and I'm just doing the same. Thank you so much for this initiative. Thank you for having setting up the presentation, inviting us.
Okay, and I hope everyone participates in the other DSA activities. If you're not part of the mailing list, let me know, and I'll add you to the wider DSA mailing list. Otherwise, have a nice weekend and the next phase of your projects, and all the best. Thank you to everyone. Bye.